I know you will enjoy hearing her tonight. Please welcome Dr. Lucie Bruijn.
Thank you, Ellen, for the generous introduction, and it's always a real privilege to be here in Philadelphia and to really have a chance to acknowledge and thank the Greater Philadelphia chapter for all their contribution to research. It's always very difficult to really mention every single study because I talk very broadly about so many things, but if it wasn't for this chapter, so many of the things would have got off the ground. So for that and for all of you and for your commitment, I'm truly grateful, and it's a real privilege to be here. It's particularly an exciting week for me. I tend to always do that, though we pack programs quite actively. I also get our scientific colleagues and clinicians quite busy because bio has indeed been a very exciting few days. I spent fewer days than my colleagues, but really meeting with industry one-on-one. And it's startling to me, actually. Often I think, well, am I going to meet any new companies? Will I know the people that want to meet with us? And in fact, we had about 10 or 12 meetings with 5 of the companies I've never heard of, all interested in starting to work in ALS research. And really building those partnerships is quite amazing and quite exciting. Then today, our colleague Dr. Maragakis has had a full day, so he's already doing his work for the day. We had a fantastic review panel, mostly from industry, to review our drug development contracts. And then tomorrow we'll have a day of bringing many of our colleagues together to talk about the projects that they are working on that are overlapping and the synergies that we can build there. So a very exciting and active week, and so happy to share with you in a snapshot, and I think, Jim, you've got to keep me on time, a little bit of all the activities. So with that, let me see. I am going to give a bit of an overview. Some of you, bear with me, might have seen it listed public policy, but I know that many of you were not here. And just a quick thing to tell you, 2 days ago I shared some of this. I got 13 minutes to present, and their forward button didn't work, and it was very challenging to fit 30 minutes into things that can't move. So anyway, the ice bucket for all of us changed the world of ALS, and I'm very committed to making sure that the dollars that we have, which are extremely generous and very valuable, will go the mile to make sure we get new treatments. I always say the perspective is it does take $1 to $2 billion to get a new drug to market. That's just a reality check, but it doesn't mean to say that with the funds that we have and the additional funds that will grow from it, that we can't get some very exciting things into the clinic. And so I hope I'll relay that to you today. So I don't even need to do it much for the audience, but I have some key points that will really come back again when we talk about other treatment options. And then I'll talk about the challenges of drug development, and actually sitting at a panel today with the industry folk, analyzing the programs on the table and seeing how difficult it is to get treatments to the clinic. It's quite sobering, and so there are things we can do, and we have to be aware of it. And then fold into that a bit of our strategy. So again, a slide I'd like to show, because this is really partly why developing treatments for ALS is such a challenge. It's not a focal disease. It's not as if you bruise yourself, you have inflammation, and you get rid of the inflammation in that local area. It's a complicated system where everything has to be perfectly interacted and interconnecting. So to solve it is you have to think of all these components. How do these nerve cells connect with the muscle, both looking at the upper motor neurons and the lower motor neurons. And more importantly, not only is it a disease of the motor neurons, but we do know in many cases there could be an additional component of frontal temporal dementia. And so that adds to the complexity of how we think of treating the disease, even caring for people living with the disease. And then a picture that can now be put into many different versions, but I still like this one. And it can go into many different neurodegenerative diseases. Actually, we could replace this with Alzheimer's and with Parkinson's. The reality is that there are many different pathways involved. Central to it is that motor neuron. But in reality, where it starts, how it moves, how it progresses, all these things come together. And in fact, we talked about it a lot today in our review of programs. What is the important target? So the target being the thing we're going to try and change with our treatment. So this is really the challenging picture we have. But it also encourages us that we can study all those various areas and focus on so many different things. So why do treatments fail? Because it's not as if we haven't tried many things. And in reality, it's that target we talk about. It's the fundamental cause of ALS. And as we know, there are some genetic causes that are the best clues for us, but a lot of ALS probably has multiple genes involved interacting with a complex environment. So what exactly starts ALS is quite tricky. So how do we choose the best target? Once we have a target and we get to treating, is that really the best approach? Is just that single approach enough? And can we actually measure it well? And some of this will come up when Dr. Berry talks about clinical trials that are ongoing and our challenges there. Do we diagnose the disease too late? Well, we know that actually, because many of you go through that pathway of very slow diagnosis, ruling out everything else. And in the meantime, the system is degenerating and the cells are dying, and so we need to get it earlier. And there are things we are doing in all of these things. We also know that ALS is not one single disease. If we look around and we know our families and friends and who are living with it and you hear, it's very variable. Some start with bulbous, some with limbs, some fast progression, some slow. So really, it's in a way like cancers, where we don't lump it all together really. We need to think much more carefully. But it's not so easy to think about how we're going to divide these groups for treatment. And there are interesting clues coming along. And of course, in reality, is one treatment is probably not sufficient. It's going to be attacking the system from multiple angles. So this is a traditional pipeline that we use in the industry. And I feel strongly, and it reflects in how we are thinking about our strategic plan, that these are some of the areas that we can really impact. Better understand the targets so that the drug you are going to use or the drugs will be more effective. Through biomarker development, which will be touched on, gene discovery and disease modeling. Improve our preclinical studies. Don't say that the animal model is useless because nothing translates, but use the animal model in context of other model systems, which are great now that we have RPS model systems, worm systems. Use the combination, but always be sure you're thinking about what is that happening in the human system. Does it really correlate? And then certainly what we've tried to commit to is improving our clinical trials. At least these are very expensive things to fund, but can we, with the smaller trials, do a better job of being sure that if we're treating with the treatment, we can really identify whether that treatment is affecting the pathway we thought was important. And again, biomarkers is key. And then together with my colleagues, Pat and Steve, we think that we can make a real impact working with our clinical colleagues and scientific colleagues to build a guidance document where we really improve and dialogue with FDA as to what is it that is important when we put things into clinical trial and develop things. So for that reason, although we have over the past been very focused on our translational program, we now have significant funds to be able to really bridge the gap between basic research in the lab, together with the academics and the industry partners, really try and push things forward. And continue to provide what is critical, the infrastructure for clinical trials, because it's done at so many centers. And consortia initiatives, many of which we'll discuss tomorrow, building teams not only in one place, through many partnerships and also globally. And not to forget that many people do ask, will we invest in people living with the disease now? Absolutely. Through assistive technologies, we're trying to explore how we can be more effective through our clinical management programs. So it's very important to do multiple things in track. And this, I'm not going to spell it through all of it, but these are the areas and the topics that we feel are high priority to develop. And this is the encouraging thing about research, actually. I started coincidentally in 1993 when the first SOD1 gene, the first gene for ALS was found. And it was a slow time before the next genes were found. But this is really, my apologies, the landscape now, where we really have multiple genes that give us real clues for that big target concept that I said, trying to hone in into the right thing. This is the picture for familial ALS, about 5 to 10% being familial. We've almost found over 60% of the genes involved there, so very encouraging, a landscape that has completely changed. The yellow chunk is the excitement around the C9 mutation, which we'll allude to a little bit more later. And because genetics plays such a fundamental part to our understanding, we've heavily invested our iSpiker funds into helping move this along. And the New York Genome Center is a physical center that was developed for multiple neurodegenerative diseases to do the sequencing. But we have helped establish the ALS part of that. And the value of having multiple diseases is really the ability to cross-identify, build good control sets, because finding new genes is easy in a way. But finding genes that are meaningful to a disease is much more challenging, because we all have different changes in our genetics that just make us our individual. But those that cause the disease, you have to delineate. So having good control sets from other diseases helps us do that. And the New York Genome Center, although physically in one space, is really meant as a global collaboration, and they will be represented tomorrow in many partnership initiatives. Project Mine is also an incredibly exciting initiative. In fact, they will be swimming in New York. I'm not sure that I'm envying that I'm not doing that on Sunday, to raise more funds and awareness for it. And this is really driven by a patient, Bernard, who was on it here 2 days ago at Bio. A person living with ALS that wasn't satisfied with entrepreneurial, wanted to do something, and really believed that if we sequence every single person living with the disease, we're bound to find all the clues. And so Project Mine, in collaboration with New York Genome Center, is doing exactly that, is planning to sequence very large numbers of people with the disease internationally, all over the world, to get the goal of understanding the genetics. Importantly, as I mentioned, I know that many companies even spoke with me yesterday and said, oh, that mouse model is useless, etc. Well, some didn't realize that I did build one of the mouse models, and together with the teams, that I have a sort of passion for the value of it, but in its place. And I do think that we need to understand its value and the value of other model systems. And what is very interesting and quite exciting, and there's some studies that will be using this, is that there is actually spontaneous mutation in a dog in SOD1, and that just gives you a different model system, bigger size, has value for various other gene therapy technologies, etc. And then again, we'll hear more today from Dr. Maragakis, the value of stem cells and iPS cells. But all of it has to be connected with what is actually happening in people living with the disease, otherwise it is rather meaningless. And this is just to illustrate, I won't actually even go into this, only to mention that we invested as part of our Ask Bucket funds into the Cedars-Sinai Center to have one of many very good core centers for IPS development. And part of the initiative is to provide these lines for other studies, and also work with teams globally to optimize how best to develop them. And then this is really a description of what we did today, actually, in the review. I feel very strongly, and partly when I came on board from an industry setting, from Bristol Myers Squibb, I really saw that there was a rigor needed for drug development that can only be done in partnership with academia and industry. And our concept here in the drug development program is try and get as many interesting ideas through that funnel. That's why we spend about $500,000 on a small contract, and see which of those can really move forward. And a great example is the antisense study. I know many have contributed a Greater Philadelphia chapter towards that. The SOD1 trial that we have a participant from, and we're anxiously waiting the next trial. And we also have a wonderful partnership with Genentech, looking at an interesting muscle concept, which is also funded through the Greater Philadelphia chapter. And these are the kind of things that are going to fill the pipeline so that we have much better agents and treatment opportunities to bring to clinical trials. So we are significantly investing in this concept and this program. I don't know how my time is doing. Okay, so this is really the antisense technology. Really in brief to say what the goal is here is that we have a mutant protein, something that is doing something we don't want in a cell. And although we can spend years, and I can spend some of my years in the lab trying to figure out what was going wrong, some thought, well, what if we just got rid of the protein or significantly reduced the protein? At least we could imagine that we could alleviate the problem and slow down the disease. And that's exactly what this technology has done. It came from an academic lab together with the clinician, Dr. Richard Smith initially, and then of course teams of others. And it was at the time a crazy idea, because actually it had never been used in a neurological disorder, and most of the time these things are attracted in fact to the liver. It wasn't clear that it would be attracted to the cells that we care about, the neurons, and in fact when that was found, the whole field has changed for antisense technology, and we now have a clinical trial for SMA, about to go in trial for Huntington's disease, and even Alzheimer's disease is approaching it. So it just shows that it could be one disease like ALS that impacts a field for many other diseases, so quite exciting. And this is my schematic to help you see that a little bit better. Basically, bad gene, block that gene, hopefully no protein. And so the way to deliver it is by two different means, either into the space in the brain or the space around the spinal cord, because that fluid is actually continuous, so it will move around where it needs to go to the different cells. So it has been in clinical trials, it is safe, we have a living person to tell us it wasn't a problem. It went back to the drawing board because with all the different diseases using the same technology, the technology improved, and so it should be, we are told Biogen INIC now is going to run the trial, it should be in trial next year, and you might hear more confirmation of that. But so the exciting thing about that investment really helped spearhead a very quick turnaround for the new discovery, which is going to use the same technology. And so it just shows that sometimes investing in technology, whether or not it's going to succeed for that particular approach, is very, very valuable. And just to tell you, many of you have heard the discussions about C9. It has a strange name because we don't know its protein product. Unlike superoxide dismutase SOD1, we've known for years what it does. C9 is just, it annotates the location where it was found. But what it has is an enormous orange triangle, an expansion, a repeat that shouldn't occur. And what antisense technology is going to attempt to do, that just shows how you can see that repeat change. Antisense is going to help block that without bothering the function of the normal protein. The intention is to stop that production of that large extra piece. Another area, and it was encouraging to see a few that will go forward for funding in this space, is that gene therapy was an area that for years actually has not been moving forward. And part of the challenge I illustrate here, and this is work from Zhu Sheng, is that the virus, which is a virus that is no longer going to infect at all, had a problem getting to where we needed it. But with various new versions, as you see, we've gone from AAV1 down to now the 9 version, we can see all you need to care about is that middle area where the green is now the spinal cord. And so before, it wasn't really possible to deliver where we needed to the things that the viral vector will take. Viral vectors and gene therapy, the concept of it is that it is a delivery system. Just like we'll hear about stem cells can be delivering things to the right place, so can viral vectors do that. And so this was very encouraging. In fact, in SMA, there's an exciting trial going on just using these kind of viruses, and it just shows again here in the brain, looking at the lower panels in the middle, it also goes into the brain. So again, without getting to where you need it, the technology is pretty useless. But this is really advanced to a point where it can really be used. And then another exciting partnership with Genentech, and driven by now funding from the Greater Philadelphia chapter and an academic partner, Steve Burden, was the concept that maybe you can improve the connection at the muscle and the nerve junction, and also improve transport, which is critical for survival of neurons. And in fact, a long time efforts and work has indicated that if you increase the levels of the kinase receptor MuSK, you might be able to do that better. And by chance, Genentech had a product on the shelf which wasn't going any further, which was actually an antibody, an agonist that could increase MuSK levels. And so just having the partnership and the resources and the reagents has spurred that field along to a point where maybe it's something that we can bring to the clinic. And I'm going to go through this quite quickly. I might be overlapping a little bit with Dr. Berry's presentation, but it's fundamentally important, and a lot of what's going to be discussed tomorrow is biomarkers. And biomarkers are quite simply said, it's an indication, like blood pressure measurements, of whether you have something going on, and then more importantly, if you treat it with something, can you change that? It also is very important for diagnosis. Of course, if you carry the gene mutation, that is a very good biomarker, early diagnostic. But in cases where we don't know a mutation, it would be great to be able to diagnose it in a much more effective way than we do now. And so I'll just go through these very quickly, but we are obviously trying to build imaging modalities in spinal cord and in brain. This is great work that actually Iron Horse was present at the Bio with their biotech company. And very simply said, it is a measure in blood and the fluid around the brain. Neurofilaments are very important in the structure of your nerve cell, and so it seems not necessarily clear exactly mechanistically why, but probably the cells are dying, and at the time that the cells are dying, This particular filament is released, neurofilament is released, and this is a ratio actually of two components, the complement as well. And all you need to look at really is at the image here, where clearly in ALS, at least compared to disease controls and healthy controls, it seems elevated. And Kevin Boylan showed together with colleagues that actually what is interesting, although it stays stable once you measure it in patients, it seems to be variable in patients, and although early, so not really completely proven, and it's what's going on in validation studies, is it seems that it seems to predict a little bit the prognosis of the disease. So the higher these levels in the patients that were looked at, the faster the disease progression. This could be very helpful in clinical trial design, because then you can try to separate people that have faster disease progression from those with slower, so we don't confuse the data coming out. And then a great study that is being done in partnership with our organization ALS Finding a Cure as part of an initiative ALS Act through the Ice Bucket Funds is the concept of personalized medicine. Not new to ALS, not completely novel to our field, but being embraced in many fields now. The concept, as I said, that we are all different, and certainly in ALS, the disease is different. And so like the cancers, we need to be better at grouping people. But how to do that is the challenge, and colleagues together with Dr. Berry and others are working towards doing this. And the study, very briefly, is looking at healthy volunteers and patients, and in a reasonable time frame with multiple readouts, and looking at many things. Imaging, PET imaging in the middle is a glial marker to look at inflammation, taking cells from the skin cells and developing motor neurons. So really trying to get a lot of information, so big data, and seeing if we can align it to make some sense of it, to be able to think about how we treat the disease better. And nothing happens without the hard workers in the lab, and we've all been there at the initial stages at some point, and it's our young fellows who really do a lot of the innovative work together with their PIs. And so our Milton Safinovitz postdoctoral fellowship is pivotal to keeping this going. We also have clinical fellowship programs because actually the clinicians are key to this. And so this is not well representing everyone, and there's always someone you leave off, but you always have to recognize that this is a team effort, and it's really great to have so many wonderful partners to do this with, many different organizations and companies. And I do feel very confident, not only because we have more funds, but because we are in a place where there is so much collaboration and there's real energy and a real sense that we are going to make an incredible difference to find, at least within the next 2 to 3 years, some meaningful treatments in clinical trials.